everyone, welcome back to Window Seat Studies. I hope you all are doing well. In last class, we were learning about X chromosome inactivation. We were discussing about stages of X chromosome inactivation, and we learned about the very first step of inactivation, and that was the counting step. So this step is crucial because it is going to determine whether there is need of X chromosome inactivation or not. So in counting, we determine the X chromosome to the autosome ratio, which is going to be the deciding factor of the X chromosome inactivation. So the counting part, the ratio of X chromosome to autosome, it is decided by the certain factors which can be either X chromosome linked or they can be autosome linked. So there is not much information available about autosome linked factors, but we do know about the X linked factors. And in last class, we discussed about such a factor, and that is the RNF12. So it is X-linked factors, mean it is present on the X chromosome. So this RNF12 is a part of the XIC, X inactivation center, and that is what we are going to discuss in detail today. So in last class we learned that the X inactivation center, all of the X inactivation center, this is the non-coding, but only the RNF12, which is the part of X inactivation center, this region codes for the RNF12 protein. So how does the RNF12 decides whether the X chromosome will be inactivated or not? That we learned in the last class. So RNF12 is produced in both males and females, but in the males, as they have only one X chromosome, the amount of the RNF12 that is produced is low compared to the females. So that doesn't reach to a threshold level which is reached in the females, and that is required for the activation of the exist long non-coding RNA. So that threshold level is achieved in the females and it activates the X long non-coding RNA. So the activation of exist long non-coding RNA, that is going to result in the X chromosome inactivation. We have not yet learnt about the mechanism of the inactivation, but we will do so in the future lectures. Today we are going to discuss about the XIC, X inactivation center. So this is a region in the X chromosome which is responsible for X chromosome inactivation. So there are certain important parts of those, and all of them are non-coding except RNF12. We have already discussed about RNF12 in the last class. Today we are only going to focus on this part. Okay. So the X inactivation center, it consists of exist long non-coding RNA. Okay. So this codes for a long non-coding RNA, and it is going to do the inactivation. Okay, so this exist is going to do the inactivation of X chromosome. There is other long non-coding RNA which is antisense to the exist gene. So this is the antisense strand, the strand which is opposite to the sense strand. Okay, so T6. Okay, as this is the antagonistic strand, as this is the antisense strand, so exist antisense strand, it is written as this. So that is why it is T6. Okay, it is written exact in the opposite manner, and that is why the T6, which is an antisense strand, so as it is antisense to exist, both of them have the antagonistic role, the opposite roles. Okay, so if the exist does the inactivation of X chromosome, T6 inhibits the inactivation of chromosomes. So whenever there will be the high expression of T6 T6 RNA, that is going to result into the active copy of X chromosome. So whenever T6 expression will be high, both of the X chromosomes will be activated. Whenever there will be high exist expression, one X will be inactivated. So there will be only X dosage, and T6 will result in 2X dosage. Okay. So these are the main parts of the X inactivation center, but there are certain other parts like FTX, JPX, DXPAS 34, X site, and TSX, which are also important regulatory regions. Okay, so the regions which are upstream to the exist, they are going to do the activation of exist, while those which are present downstream to the exist, they are going to do the activation of T6. Okay, so this is just for remembering. This will help you to remember. So FTX and JPX, both of them, they activate the T6 exist RNA, while the DX pass 34, excite and TSX, all of the three, they are going to activate T6 RNA. So we can say that uh, we can easily say that the FTX and JPX, both of them, they are involved in inactivation of X chromosome. Like they are going to do the inactivation of X chromosome, they are supporting inactivation, while the DX pass 34, excite and TSX, all of those three, they are going to inhibit the X chromosome inactivation. So they are going to activate the T6, which is going to support the 2X dosage. So this is it about the X inactivation center. So there's one more thing that I would like to add here. Okay. So in last class we learned that in the first class of X chromosome inactivation, we learned that the pluripotent cells, they are somehow linked with the 2x dosage. Okay, we learned that this in the first lecture, that there is some correlation in the pluripotency and both activation of both X chromosomes. So what happens is that in the pluripotent cells, those cells have some factors known as pluripotency factors. Okay, so the pluripotency factors such as OCT4 or NANOG, all of them, they are going to lead to the reparation of RNF12. Okay. So as they do the reparation of RNF12, so now RNF12 won't be there, and so it won't lead to the activation of exist. Okay, so there is no activation of exist, and OCT4 and NANOG, they also activate T6. Okay, so they do the two jobs. First, they repress the RNF12 and again they activate the T6. So now there will be no activation of exist. Instead, there will be activation of T6 RNA. So that is why, due to this, there will be no X chromosome inactivation, and the pluripotent cells, they will show 2X dosage. So this is why pluripotent cells, due to the pluripotency factors, the Yamanaka factors, OCT4 and NANOG, they show the 2x dosage. So so this is how the this is how the counting is made, and this is how it decides whether the X chromosome will be inactivated or not. So after the first step counting, it comes out that there is indeed need of the X chromosome inactivation. So that, let's assume that that cell is female, so it has two X chromosomes and there is need of X chromosome inactivation. Okay, so once the counting is done, the next step in the X chromosome inactivation is choice. So now you have decided that you have to do the X chromosome inactivation, but now you have to make the choice that which X chromosome is going to be inactivated. So the next step is the choice of the X chromosome inactivation. So this choice is of two types. The choice could be random or the choice could be skewed. Okay, so we learned in the last class that the random X chromosome inactivation, it is seen in the placental mammals, but the extra embryonic tissue like the trophoectoderm, which is going to form the placenta in future, only that part of the placental mammals shows the imprinted X chromosome inactivation. Okay, this is what we learned in the last class. So in imprinted X chromosome inactivation, it is already decided that either paternal copy or the maternal copy of the X chromosome will be silenced. But in the random X chromosome inactivation, the choice is very random. So like you can't pinpoint that this X chromosome will be inactivated or that chromosome will be inactivated. So let's assume that there are two rods, okay, and both of the rods are sticky, and those sticky rods, we are placing them in the sand. Now as the rods are sticky, the sand grains will stick to them. So when you take the rods out of the sand, a lot of sand grains will be stuck to that rods. So when you bring those two rods together, those sand grains, they will be redistributed on the rods, okay, and that redistribution will be random because you don't know, both of the rods are equally sticky, so the redistribution of sand grains, it will be random. You can't pinpoint that this rod will have more sand grains or that rod will have more sand grains. So this is how random inactivation of X chromosome is done. Like the the sand grains that I mentioned are the X-linked factors, while the rods are X chromosome. So the redistribution of X-linked factors on those X chromosome, it is random, and when it is random, the X chromosome the inactivation will also be random. So this is the random choice, okay, and this is the widespread phenomena. The random inactivation is quite common, but there is also a phenomena known as skewed choice. Here there is some biasness. So like assume that instead of making both of the rods equally sticky, you show some biasness and you make one rod more sticky. Okay. So now one rod is more sticky, so this rod has more probability of of having more sand grains. So this rod will have this has the higher probability of having more sand grains. And now as it has more sand grains, means the X-linked factor, it has higher probability of being inactivated. So this X chromosome, the rod that we are here, the rod symbolizes the X chromosome. Okay, so this X chromosome, which is skewed, which is more sticky, that will have the higher probability of being inactivated. So this is how skewed choice is done. So this skewed choice results in certain syndrome. So there's a syndrome known as Rett syndrome. Let's discuss that how the skewed choice leads to the Rett syndrome. So this is a neurodevelopmental disorder. This is a neurodevelopmental syndrome. Okay, so what happens is that in the Rett syndrome, the protein MECP2, this protein is mutated in the neuronal cells. Okay, so this is mutated in the neuronal cells. So the mutation of MECP2 leads to the Rett syndrome. So let's take a look at how the skewed choice is going to result into Rett syndrome. So assume that the capital M, this means that this is wild type genotype, means the MECP2 will not be mutated, while the small m means mutated MECP2. Okay. So now let's assume that the embryo has the genotype MM. So it has one copy of wild type and other copy of mutated MECP2. So let's assume that the embryo which is formed, it has the wild type copy from father. So the wild type copy is the paternal copy, while the maternal copy is the mutated copy. Okay, so this is the genotypic constitution of embryo. This is the genotype of embryo. Okay, so now the X chromosome. So this is MECP2. It is present on X chromosome. Okay, so this is MECP2. This is X-linked gene, and that is why the X inactivation is going to result into Rett syndrome. So it is present on X chromosome. So if the wild type copy of the if the X chromosome on which the wild type copy is present, if this gets inactivated, the embryo will only be producing the mutated MECP2, and that is why it will develop Rett syndrome. But if the X chromosome, which have the mutated copy of MECP2, if this gets inactivated, so this pink one, this pink X chromosome gets inactivated, it will not lead to the Rett syndrome because you have the wild type copy which is present on the other X chromosome. Okay, so if this gets silenced, it will lead to the Rett syndrome because only this copy will be present. And if this gets silenced, mutated one gets silenced, there will be no Rett syndrome. Okay, so I hope this is clear to you. So now let's see how the skewedness plays a role in the Rett syndrome. So let's assume that the chances of the silencing of paternal X chromosome like this is more in case 1. Okay, so in the case, there are two cases, case 1 and case 2. In the first case, the probability of silencing of the maternal copy is more, and in this in the second case, the silencing of paternal copy. Okay, so let's assume that in the first case, this copy, the probability of silencing of paternal copy is more, while in the second case, silencing of the paternal copy is less. Okay, the probability that this gets silenced is higher. Let's assume this is 80%, and probability of this the paternal copy being silenced in the case second is less. Okay, so now if there is some skewedness in X chromosome inactivation, let's say in the first case, the X chromosome, the paternally inherited X chromosome will be inactivated. So the case first is paternal imprinted silencing or inactivation of X chromosome, or the case 2 is maternal imprinted inactivation of X chromosome or maternal imprinted silencing. Okay, so we that is already decided that there is already skewedness, that either paternal copy will be silenced or the maternal copy will be silenced. So in the case one where the paternal could be silenced, so this copy could be silenced. So there are the higher chances of Rett syndrome in the baby. So the probability that the baby would develop the Rett syndrome, that probability will be higher. So higher chances of Rett syndrome will be involved if there is paternal imprinted silencing. But if there is maternal imprinted silencing, the chances that the embryo is going to develop Rett syndrome, that will be low. So here the Rett syndrome, the baby having Rett syndrome, that probability will be low. So this is how the skewedness of whether the paternal copy or maternal copy will be silenced, that plays a role in the in the developing of the Rett syndrome. So this is it about the choice part. In the next class, we will learn about the next step of the X chromosome inactivation. We'll see how the X chromosome inactivation is initiated, how the X chromosome inactivation is established, and how that is maintained. So those are the next stages of the X chromosome inactivation. We will learn about that in the next lecture. Till then, see you. Bye bye.